Forensic Science Lecture 9-3, Principles of Heredity. So the ABO versus the DNA when it comes to identifying an individual um, um, has become much more advanced with the DNA. A, B, and O typing is still very important, and prior to any sort of DNA analysis, blood stains were linked to a source using the antigens, blood proteins, and enzymes for the ABO typing. This, however, has been replaced with modern DNA technology. They still will do the ABO, but they're not using it to link it necessarily specifically because it's not quite as accurate. But the DNA technology allows forensic scientists to link blood and semen stains to a single individual down to the pinpoint. When we're looking at heredity, all antigens, proteins, enzymes, they're all genetically controlled traits. They're determined. The identification of traits can actually, or determining what an identification of the trait can actually provide insight to how one individual compares to another. It also is how, or shows how biological substances determine an individual's makeup, and it allows us to see how they are passed to future generations. Couple terminology that you need to refresh on are things like genes and chromosomes. So remember that the basic unit of heredity is a gene. Each gene controls a specific characteristic, and the genes are positioned on chromosomes. Remember, the chromosomes are the long threads of nucleic acids that are found in the nucleus. Almost all human cells have 46 chromosomes, or 23 pairs. The reproductive cells are the only exception, with 23 unmated chromosomes. So the reproductive cells, remember, are the sperm and the egg. During fertilization, when the sperm and the egg join, they form what's called a zygote, which is the fertilized egg with the correct number of chromosomes. So a zygote now contains 46 chromosomes, or 23 pairs. Each gene occupies a specific location on a chromosome. This is known as its locus. Genes are determined that determine the same characteristic, occupy the same locus on all human chromosomes. Remember, alternate versions of a gene are known as an allele. You have either a dominant allele. That means that that trait will show if it's present, and then a recessive allele, which are hidden if a dominant is present. The only way that a recessive allele can show is if both alleles in the individual are recessive. If the alleles of the same gene are the same, they're said to be homozygous, and there are two forms of that. If there are two dominant alleles present, they're considered homozygous dominant. If there are two recessive alleles present, then they're considered homozygous recessive. And then if there is a different allele for the same gene, for instance, if one is dominant and one is recessive, in the combination, then they're considered to be heterozygous. In some cases, there are more than just one dominant and one recessive. There may be multiple alleles that are dominant, and in some cases, two dominants that pair up both show, and this is considered to be codominant, which is what we see in our human blood typing with that AB blood. When we look at the chromosomes or the allele pairs, they create the genotype. There's no known lab test that can determine a blood type's genotype. The person's outward appearance is its phenotype. You can't always tell what the genotype is by a phenotype, especially if that dominant trait is showing, because it could be masking a recessive allele. However, genotypes do determine the phenotype. And we can use Punnett squares to help determine a person's possible genotype. And so if you have two parents and both, let's say, are possible heterozygous, you can create a Punnett square, which is the box with 4 individual boxes. You place the parents' alleles, one parent allele on top, one parent allele on the side, and then much like a multiplication chart, you fill those in. And those are the possible outcomes for every given cross. So every time these two parents create an offspring, they have these possible outcomes. It's not saying that if they have 4 children, 1 is going to be this, 2 is going to be another genotype, and there's a 3rd genotype, or what have you, however it comes out to play in the Punnett square. It's every time that they have an offspring, these are the possible outcomes. When we're looking at genotyping of blood, there's actually no direct relevance for a criminal investigation, but blood typing plays a big role in paternity cases. Eggs only have the female sex cell only carries X chromosomes. 2 X's are the chromosomes for a female. Sperm, on the other hand, carry either an X chromosome or a Y chromosome. So ultimately, it's the father who determines the sex of the offspring. As they are blood typing, the more blood group tests that they do, the better the chances of excluding a male, or if there are no discrepancies found between the male and offspring, the more certain that that male is the father of that child. But paternity tests often involve more than just ABO factors. They also contain HLA factors, which are human leukocyte antigens, which relies on antigens of the white blood cells. If the tests cannot exclude a suspect, the chances are better than 90% that he's the father. Now that labs have implemented DNA testing into the paternity test, it actually raises the odds of establishing paternity above 99%. So paternity tests nowadays are pretty accurate. It's hard to kind of deny it if they comes out positive or negative. This is the end of lecture 9-3. There will be a quiz next class.